Hello, mate. What are you doing here? You've taken me by surprise. I was expecting that Gwilym Roberts bloke to turn up, at least virtually, and I've got you in person sat next to me.
He's too busy for us again, Lee.
What's he doing? Do you know what he's doing?
I've no idea what he's doing, actually.
Some kind of high-level international engagement stuff, I would have thought.
It always is, isn't it?
Bit of a kind of secret scribbly kind of character, isn't he, in that regard? It means I can't have my normal kind of backstory chat that I do with Gwilym because we see one.
I haven't been baking bread. We can talk about your tortoise if you want.
Still hibernating at the moment, okay. So he's still tucked away. So what exciting things are you doing at the moment? What's big on your radar?
Well, I've been working on our annual report, which we're about to publish. I think when this pod goes out, we'll be publishing our annual report on the same day. So, yeah, I've been looking back at our successes over the past year, our big achievements.
We should probably do an annual report podcast, shouldn't we? Not thought about that, but we should probably do that. That's probably. Have you had any hold-ups in the production of the annual report? Any kind of content bit late in coming in or anything like that?
Rather not answer that question.
That's me.
No, I'm incriminating myself.
Incriminate me.
Well, yeah, you've been a bit.
I've been a bit lax, haven't I? Yeah, but it's all up together then, and so we can look forward to a really kind of nice glossy annual report in a week.
Absolutely.
So, yeah.
I mean, one of the things that I've been looking back on was our success over the past few years in the Earthshot Prize.
Ah, cool. So that's a nice segue into the podcast today, isn't it?
Seamless, isn't it?
Absolutely seamless.
Lee Davis and Gwilym Roberts are the two IPs in a pod, and you are listening to a podcast on intellectual property, brought to you by the Chartered Institute of Patent Attorneys.
Jim, are you there in the background?
Yes, I am.
Told you it was going to be a load of waffle that you can just listen in on. Introduce yourself to our listeners, if you would, if that's okay.
Sure, I'll give a short introduction. Yeah. So I'm Jim Ajioka. I'm the Chief Scientific Officer for Colorifix. The whole enterprise kind of started in my lab several years ago. Ori Arconi was a postdoc in the lab at that point, and he's now the CEO of the company. And frankly, he's probably more important than me at this juncture. But yeah, so I've been working on making color and doing sort of engineering biology for about the past 15 to 20 years. So it's kind of nice to see something that I worked on as an academic in my lab in Cambridge coming to some kind of reality, and it's been a really great journey in that regard.
So yeah, we can meander through that. Before we do, I've been learning a little bit about fabrics and colour recently, but through the lens of historical podcasts that I listen to. So bear with me, Jim. There is a point in this. I learned on the treadmill when I was running yesterday that the Romans used to dye their fabrics largely through natural available dyes by kind of crushing up shells, which was a Tyrolean red colour, as I understand it, and human urine was a big feature in dyeing back in those days. Tell me we've come on some since then, please.
Yeah, we have, but the principles are pretty much still the same. You try to find a color that will stick to a fabric, won't fade in light, and pretty resistant to washing if you wash your clothes. And there are different kinds of methods to make the pigment either change color or stick better. Probably the urine is doing that. So, it has come on, but the principles are pretty much still the same.
So what were the challenges that you guys tried to overcome then? And so as I understand it, for a long time, the fabric dyeing business has been quite synthetic and harmful to the environment and all of those kinds of things. And you've come across ways of working that change that, yeah?
Yeah, well, maybe I should just give you a bit of a historical background on the chemical textile dyeing industry. So back in the mid 1800s, the empire in this country anyway was at quite a height, and there was a lot of people that were dying from malaria in the tropical areas of the empire. And so there was a chemist named William Henry Perkin that was trying to synthesize quinine, because that was the drug of choice to fight malaria. And the only way they could get it was from a bark of a tropical plant, and that wasn't readily available. And other countries controlled that market, so it was really difficult. So they were trying to make a synthetic malaria treatment, but he failed at that. But what he did do was make a really nice purple dye called movine. And it's the first aniline dye, and it became something really useful during that period of time. So people wore a lot of purple clothes Purple's always been kind of a royal color anyway, starting with the Romans. So it became really popular. At the same time, the Germans discovered the same thing. The German chemists were probably the world's leaders at that point, and they made a similar discovery. So, the company BASF, it's a big chemical company in Germany. Well, the A in BASF stands for aniline. Okay, so they were making a lot of their original products were chemical dyes, and they continue to do so today. So there's been a long history of trying to make chemical dyes from petroleum products, and it became a lot cheaper and easier to do that, and it could scale. Okay, one of the problems with traditional dyeing is it doesn't scale very well because you have to make extracts from plants or insects or rocks. You just can't make enough of it quickly enough to make it a commercial entity. So it really mattered that they were able to make these synthetic dyes, and eventually with chemistry you can make all sorts of different colors, and you can make them perform in different ways, have better light fastness characteristics. Now they have dyes that will covalently bind to the fabrics themselves, so the reactive dyes. So there's a variety of different improvements that have happened over time. But one of the things that really hasn't improved that much is the kind of dyeing process itself with regards to the chemicals that are needed to do it, the dyes themselves, but also the efficiency of the process. So if you look at the chemical dye industry, you have to, and we can talk more about the different types of fabrics and what happens, but when you look at the chemical dye industry and textile dyeing, there's probably about 200,000 tons of excess dye that just gets wasted and put into the effluent because the process is so inefficient. Okay, so think about 200,000 tons of dye just going into the water. Okay, that's not really a great image in my mind. Also, it uses a lot of water because it's inefficient. The washing that you need to take out the excess dye and make it into a point where the fabric is dyed to the extent you want, but no more of the dye washes out. It can be really, really extensive. Whereas with our process, we wash once and we rinse, and that's it. Okay, so our process is a lot more efficient, which is part of the beauty of it. Okay, we also leverage biology in a couple of different ways that we can talk about more specifically, particularly with regards to our intellectual property or our kind of patented method for doing the process. So I guess when you look at overall, we can save a lot of water. 5 trillion liters of water are used annually to dye. It's a massive amount of water. Talked about the chemistry that's involved. It's really unpleasant. And so as a consequence, a lot of the energy and greenhouse gases that are produced in the meantime are high. Okay, so why don't you guys, I've talked enough for a little bit. Why don't you guys?
Well, Jim, I'm just interested in, you talk a lot about water and effects on water and drinking water.
Yeah.
Didn't you guys come up with the idea when you were monitoring drinking water were you working in Nepal like 8-9 years ago?
My position at the university was in the Department of Pathology, okay, kind of oddly. And so one of the projects that was going on was to look at lots of disease factors in countries like Nepal and Bangladesh, places where the medical infrastructure isn't as kind of well-funded or as stable as it is here. So as part of this project, we wrote a grant to look at arsenic poisoning in that region. Okay, if you look at valleys that are below new mountains, i.e., things like the Himalayas or the Rockies, they're often laden with arsenic just for geological problems. Okay, so if you live below the high mountains, often your groundwater, if you use well water as your source, is highly contaminated with arsenic. So I have friends in California, for example, that have houses that are below the Sierras and they can't drink the well water because it's contaminated with arsenic. So it's everywhere where there's new mountains. So it's not, but the problem with places like Nepal and Bangladesh is that they have their surface water is so contaminated because because of farming and everything and human activity, that they can't really drink the surface water because it's so contaminated with pathogens. Okay, so back in the sort of 80s and 90s, they started drilling a lot of very shallow tube wells where they basically jam a pipe down in the ground, and eventually they hit an aquifer that they can pump the water out of the ground. Unfortunately, those aquifers are laden with arsenic. Okay. And so we were trying to make a device or engineer a device using biology to be able to monitor each of the wells, because the aquifers themselves are very scattered. So some of them will have arsenic, and maybe 100 yards away they'll be clean. And so so you have to really monitor every well to make sure that you're drinking clean water. Now, if you go back to Kathmandu, where you're in the mountains essentially, they don't have an arsenic problem there. But when we were working with the NGO that was guiding the project down in the Terai, they're based in Kathmandu, and so we said, well, why is the water so bad here? You don't have arsenic. And he said, well, the textile industry just dumps their waste straight into the river and into the waterways, and so it's just really toxic. So anyway, we were in Kathmandu seeing this and said, well, maybe we can do something. We were making colors anyway for other things in my lab. So I said, well, maybe we can figure out a better way to solve this problem, because the people, like most things in life, if the people that suffer are the people that can least afford to do anything about it. And so, we were kind of compelled by that, and just seeing what was going on firsthand to think maybe we should try doing something different about it. And because the arsenic project got, well, let me put it this way, Brexit kind of killed it for a variety of different reasons, that we decided to launch into this instead. And so then we started doing some experiments in the lab, and eventually got to the point where we thought we could have a process that we could patent. And that was really probably the start of Colorifix.
When did you recognize that you had something that needed to be protected? When did the IP bit become important?
Yeah, okay. So engineering biology has lots of different facets to it. And, for a very long time, we've been able to, put pieces of DNA into other organisms and make it do something, okay? Generally, this has been done in microbes and bacteria. Yeast has kind of been the workhorse organisms for sort of genetic manipulation, plants as well, but mostly microbes. And so it's really hard to understand how you're going to get IP on certain things, like, being able to, make a protein, for example, in a microbe that maybe normally is in a plant, for example, because it's really easy to get around any patent that you might try to put on those things. So you start thinking, well, what are we going to do to try to, control our process and control anything that with IP to be able to start a company. And so we thought, well, maybe a process patent would be better, because that's something where people don't really understand how it works, or they haven't, put a lot of effort into, when we scanned the patent literature, there was nothing in it, literally. And so we thought, well, maybe this is where we can start, because process patents are good because they're broad. They don't patent the organism itself, and therefore you have a better chance of making sure that somebody isn't going to tell you you can't do what you want.
Yeah.
I don't know how you guys feel about patents, but most of it's about freedom to operate, not keeping other people out of the market, because inevitably that will happen. But you don't want people to stop you from doing what you think is useful.
Yeah, absolutely.
And so that's what we did. And we did enough experiments in the lab to show a convincing case that we could do this reproducibly, and that it had real commercial value. Then there was some novelty to it, because essentially people hadn't done this before. Having said that, when you look, and this is something that people should, if they're getting into the biological, or engineering biology space, they need to think beyond just a plant or microbe to make stuff for them, to produce stuff for them. They need to leverage it more than that. So in our case, we're leveraging the microbe's ability to make the pigment, but also to deposit and fix it onto the textile. Okay. Now, I don't know about you guys, but I've lived in this country now for more than 30 years. And unlike Southern California, if you leave your tea towel on the side, it's probably going to rot. Okay, or it's really dry in Southern California, so we never have mold and mildew issues, right? But you do in a big way in this country.
Yeah.
And is left on your tea towels are these nasty black spots.
Yeah.
Okay, it's a microbe, it's a mildew, it's a yeast making us a fungus. It's making this black pigment that's then deposited onto your tea towel, and it's really hard to wash out. That's what we do.
I've never thought about it that way. That makes sense.
That's kind of, yeah.
I was just going to ask you if you could break down your process, but you've just kind of beautifully summarized it there, I guess.
If this were rocket science, I certainly couldn't do it. It's just how well you observe, right? So a lot of biological science is observation anyway. So we're good at seeing things. Our biologists are, okay.
So the challenge for you, if I've got this right, isn't necessarily the kind of organic chemistry, biology side of it. That happens. Is the challenge getting colors right? Is the challenge?
Yeah, the engineering part of it comes in mostly with trying to make color, okay. And being in here in Cambridge, where DNA sequencing was really kind of, at least the conventional stuff was invented. And even the current processes that are big are still being used. We're also people in Cambridge that developed them. So we see a lot of Sanger Institute. So I've seen a lot of DNA sequence come out over the last 30 years. And it's been just absolutely spectacular, just seeing how all these different organisms have different ways they've arranged their genomes, but also all the genes that code for proteins that will be able to be enzymes to build things. Okay, in this case, colors. So what we can do is we can identify a pigment, and a lot of them are already known, like indigo. Okay, indigo is a really well-known color. Okay, everybody does it. We're working on it too for a variety of reasons. But indigo, if it can be made from an amino acid with a single enzyme. Okay, so it's a really good target to work on. Other people have done this too. We're not the first, right? But trying to figure out how to do it in a clever way is different. But anyway, so what you can do is to go, oh, I like indigo. Well, what's the precursor to make indigo? Well, it's an amino acid. Okay, it's one of our protein building blocks. Okay, so you say, okay, what do we need to convert that amino acid into indigo? Well, it's a single enzyme. Okay, so we can find the DNA sequence for that enzyme, put it into a microorganism, and it will take the amino acids which are in the cell anyway and convert it into indigo. Okay, so it's in certain ways it's a very straightforward process because most of the precursors are things that are in all cells anyway. Okay, so the process. Yeah, go ahead.
I was just going to say that you use genetic modification processes then. And that's had a bad press over the years and understandably holds a bit of fear for many people. But I guess society has got to get its head around what GM processes can actually do for the environment and for our future.
Yeah, I think that's true. But for us, we probably are in a slightly easier space because although our process is GM, our product is not. Indigo is indigo. It doesn't matter, right? Whether you get it from a plant, whether you synthesize it with chemistry, or whether you make it like we do using a microbe as a surrogate producer. So I think people are a lot less fearful of that. And, in the way our process works, all of the biomass that isn't the indigo essentially gets washed out. So, there's nothing left that's GM. People do need to understand that, it's like every other technology. There are definitely advantages, but there's also things that you need to be cautious about. I mean, I spent 30 years as a biological safety officer in the Department of Pathology in Cambridge. Okay, that's where you see a lot of nasty potential things happening, and so I've thought about some pretty horrible things over the years.
I can only imagine. Can we go back to the IP journey a little bit? So at some point you would have thought to yourself, need a patent attorney. Should we give your patent attorney a shout out? And how did you find them? How did you pick a patent attorney?
Okay, so our patent attorney is Craig Titmus. He works at Mathis Squire in Cambridge, and we found him. So we looked at a number of patents and kind of got the format, figured out what you needed to write into them, just kind of made ourselves a little, sort of outline of what we needed to do, and just went about, finding the appropriate literature to cite, the appropriate patents that were there, in place, because scanning the patent literature is really easy now compared to the way it used to be.
Yeah.
And then scanning, the scientific literature to make sure that things hadn't been published previously, and we just wrote it up ourselves for the most part. For that particular one, Craig didn't have to do a huge amount except for make sure that he closed all of the issues, and we had several conversations. But we've done a lot of work with him since then. I mean, you can write the patent and get it granted in one place, but then going international is a completely different issue, because you have all sorts of different territories you have to cover, and the way they get covered is it's almost a Byzantine maze. It's really difficult to navigate.
So the strategy of patenting was as important, if not more important, than the process. Yeah.
Yeah, I think so. I mean, so just going about, and besides, you learn a lot of things. So we have another patent going on our hardware, which is another thing maybe we can touch on. But so you learn a lot about doing it once, so doing it the second time is somewhat easier. And again, we wrote up most of what was necessary, but because we're not hardware specialists, Craig did a lot of work on that one. So we're continuing on that. So that one's pending still. I don't know. Does that resonate at all with you guys? I don't know. Maybe it's because
I suppose we should just add to that. I mean, it's interesting, your view and your thoughts on that, but we should just add that I think less than 5% of unrepresented applicants actually get their patents granted in this country. I think that's...
Yeah. You need to have an attorney.
You need to have an attorney with you.
Yeah. I'm not saying that you shouldn't have one. Yeah. What I'm saying is you can do a lot of the work yourself. Okay.
Sure.
And that's useful. I mean, my dad was an engineer. He had like 30 or 40 patents himself. So I had listened to this all my life, right? so trying to do it DIY, it wasn't frightening in that way. Anyway, so back to the thing. I think, people should, if they're going to want to patent stuff, they should try writing it themselves, at least in the first instance, because one, they'll be able to communicate with their patent attorneys much better about what it is they're trying to achieve, because they'll understand the process of novelty, inventiveness, all those sort of things that are really critical to making it work. What do you guys think? This is really interesting. I should ask you guys, what do you think? Because we just DIY'd stuff. I have no idea what real people do.
Go on, Neil, you're best at answering that.
Well, I mean, I've just spouted a stat at you, Jim. So I think that stands on its own, doesn't it?
Yeah, it does.
It's also worth saying to any people who don't know really what the background of patent attorneys is that they're all scientists by qualification and background. Yeah. So they are all specialists in their own right.
Yeah.
We're trying to find one that's a specialist in here. You have to be a specialist in both textile stuff and in microbiology.
Yeah. That's it. That's a unique combination.
That covers those things, right?
But that's what you're there for, isn't it? You're the expert in those fields. The role of the patent attorney is to take your knowledge and make it stand up to the legal test.
Yeah, no, that's right. Don't get me wrong. You need an attorney. If anybody's listening, don't listen to me saying you don't need an attorney. You do, okay, and you need a really good firm, okay. But what I'm saying is, I think it's instructive for both parties and facilitates the interaction a lot more if you really understand what you're doing, okay, or what they're trying to do on your behalf.
Would you say it's like a collaboration?
Yeah, absolutely. If it's not, then you're probably not doing your work, your homework. Yeah, it definitely should be a collaboration.
What if any hurdles have you faced along the way, either in terms of the technology or the IP side of it? Have you found it? Have there been any barriers?
Well, there's been lots of different ones along the way. Okay, so when you look at our process, if you're an engineer, okay, biologists love variation. We love nature. We like to see how the multiple varieties of trees and plants and animals, right? If you're an engineer, you hate variation. You want to make it work the same way every time, all the time, right? So there is a kind of a conflict in, it's almost an oxymoron, engineering biology, okay? It's almost an oxymoron because biology doesn't really lend itself completely to engineering. But you can put engineering principles in place to help you guide your process, okay? And that's something that we do that a lot of, kind of people that claim to be biotechnologists or, bioengineers don't do, okay? And I think therein lies the difference between us and them. So that's a hurdle, because getting people to understand why you're putting in a design, build, test, learn cycle into everything that you do. And if you're an engineer, you go, oh, that's obvious. If you're a biologist, you're going, what? They don't understand that. When you're building, it's an iterative process. It's not like you have some instant, clairvoyant moment where you go, oh, this is the right thing, and then it's instantly that. If you're an engineer, engineers laugh at you if they think that's actually how things work. Yeah, so that's a hurdle, because you're trying to get people that are in for funding, for example, getting people in pharma, or people that know how to fund pharma or fund other kinds of biotechnologies, to understand how you are going to be really engineering the process. And it's not just a bunch of random, trials of doing this or that or the other thing. When we take a color pathway, you start out building one that's kind of the rudimentary skeleton thing. You test it, does it make color or not? That's one thing. We have a really good assay. It's like, did it make color or not? You just look at it. There's none of these complicated spectroscopy or anything. It's like, did it work or not? Which is fun.
Does it look nice?
Yeah, it doesn't look nice. And then you have the other tests of, does it light fast? Is it wash fast? Is it, rub fast? All those sort of things. But, in the end, what you're really trying to do is just to say, did it make color or not? Okay. And is it a color that goes onto fabrics? So it's really fun and obvious what you try to do. So
do you always know what color you're going to get? Or
no, and this is the other thing. So there, almost every time we've done this, we're going after color, blue, right? And and you can make it, and when you grow it on the plate, the bacteria is nice blue color. And then you put it on the fabric. It's like, why did it go pink? and part of it is that, and this, if you're a real textile dyeing person, they know this too, that what one dye will do on one fabric will not respond the same, because it's all an interaction between the dye and the fabric or the surface, and what colors, what part of the spectrum it absorbs or reflects is dependent on both things together. And so, so you get some fairly radical changes, and also things like the pH matters. So, if it's acidic or if it's basic, it can hugely change the color. And so instead of being a problem, it's just a new opportunity. Oh, we've now made a different product, and we get 2 for 1, right? So it's fun.
What's the future strategy? You guys scaling up? Is this going to be you against the world, or are you licensing? What's the way forward?
So when we started looking at this, you realize you don't know certain things. Okay, we knew nothing about the textile industry. Okay, that's not a great starting point for somebody that's going to. Not great. So if if you have even half of your wits about you, go, we need to find a man who knows. Okay, so thankfully, he's Portuguese. So Portugal's one of the very few places. Well, it's probably the only mainland Europe place that still has a textile dyeing industry to speak of. So we could, find experts there. So we found a guy, Rui Sa, who liked what we did, but he's been in the industry for 25 years. So he knows how the whole thing works. He knows how the brands work, how the mills work, how the dye houses work, how the chemical suppliers work. So he knew the entire, supply chain and everything, and the business aspects of it too. Yeah, this would never work unless you actually had somebody that knew what they were doing.
And was it easy to recruit, or was he just excited by what he saw?
Yeah, we were trying to, talk to mills and dye houses, and he happened to be one of the people that was hanging around when we were discussing this, going, wow, that's really cool. I'm sick of doing what I'm doing. I'll join you guys. So yeah, he's great, because he's like Mr. Get Stuff Done, and he's just highly energetic. We can talk about the hardware, which he is also instrumental in designing. I mean, okay, so I think I left the problem back at the point at which, engineers like to have things work the same way all the time. Biology doesn't work that way. But biology, it's really the case that if you have the same genetic crop, okay, and you have the same food and the same, place you're growing it, you'll get a consistent thing. So food and environment and genetics have to be stable all the time. And what you find is that food varies quite a bit in biology. If you buy it from different suppliers, the same thing is claimed to be the same. Bacterial food isn't the same, because you'll get different performance characteristics out of them. Okay. So you have to standardize everything. So standardizing the food, and in this case, the environment, meaning the vessel in which you grow the bacteria, has to be the same everywhere, or you won't get the same product. And color's your product. In our case, the product is the ferment, so it has to look the same. There's no downstream processing. It has to look the same all the time. So we had to make our own bioreactors, and we had to make our own food. Okay, which either one of them are massive undertakings in of themselves because of the supply chain issues. We started doing this in COVID, and we hit things like, oh, we can't get the vessels made because there's no stainless steel. Also, like everybody else, oh, there's no chips, which means your control panels are not being made. So, you face all these. So we're doing this in the background of just trying to do the engineering biology end of it. So we're just juggling all these things, trying to make it work. But now, we've set up shop in Portugal to make the bioreactors, and we've set up shop now in India to get a manufacturing partner. But the other thing about bioreactors is that they're generally made for GMP facilities or places that are really clean. Okay, they're meant for pharma for the most part, and so they're very usually kind of bespoke machines that have to be set up in very specific places, and they take a long time to implement. We bought one that we thought would be just kind of a sort of control bioreactor, conventional model, and it took them 6 weeks to install it. Think about that. Ours are plug and play. You roll them out of the container, put them on a skip, take the forklift, unload them off the truck, plonk them down, hook up water, electricity, and steam, and some drainage, and you're ready to go. So when we did this with our most recent bioreactor in Cambridge, it took us 3 days, okay, versus 6 weeks. So, the idea is that because you have to do this fast, but fortunately dye houses all have steam, electricity, and gas and water. So they have all the services there already, which is because they work with high pressure vessels anyway, because all the dye machines work that way. So it's a good environment in that way. The downside of the environment is really dirty. Okay. So GMP facilities are HEPA filtered. They got, it's really clean air going in and out. Just think of the worst men's room you've ever been in. It's kind of, that's what dye houses are like, right? They're dirty, they're nasty.
They're still using human urine. Is that the
Yeah. Well, let's not go there. But yeah, they're not clean places is the point. There's lots of mold and mildew and viruses and fungi and stuff running around.
Yeah.
So we had to invent a new way to clean the air, which is one of the things we did, because the filters are these kind of membrane HEPA filters, and they clog really fast. And so we had to invent that. We also had to make a machine that you could clean in place, because you can't dismantle the things, because our turnaround time's about half to maybe a quarter of what normal bioreactors are. So you have to be able to make your ferment, clean the tank, turn it around, and start a new thing up, and we can do it in about 2 hours. Okay.
So I mean, what I'm hearing is that the business model is as much around the hardware as it is around the technology.
Well, it is at this point, because it's in the end, it's about standardization and making sure that you have a product that's real. Okay.
Yeah.
But it turns into part of the business model as well. So, we'll sell the bioreactors at a reasonable cost. We can also make them, like we bought a 30-liter one from a regular manufacturer. It costs us about 130,000 pounds. We can make ours for maybe less than 100 euros. Okay. So, and they're 10 times bigger. Okay. So we can afford to do things that normally people can't do. And so you pass that savings on to your customers, who are the dye houses. So we license the technology of the dye house. We don't sell them anything, because if you sell your microbes, then they own them and they can do what they want with them. Yeah. Obviously, that's bad for your business. So we use an MTA model. So we just use an MTA, put that in place, so they can't sell them, they can't distribute them. So it limits a lot of what you would normally be worried about in business. And we don't bother patenting them either because there's no point in it. Almost everything is easily gotten around if you think a little bit about it. We don't guarantee anything unless you're using our bioreactors in our food. So, so it's kind of a inkjet printer model. You just sell the printer at a reasonable cost. And then you just keep selling the ink, in this case, just the food.
Can we talk a little bit about your impact on the environment and the recognition that you've had via Earthshot Prize, and you won other awards, I know.
So
you've been able to quantify it.
Yeah, I mean, you can. Okay, so we've done an LCA analysis. Okay, this is part of what I think made people think that our entry into Earthshot was worthwhile. Okay, because it's really easy to hand wave, and there's a lot of greenwashing in the kind of fashion and textile sector, and we didn't feel like that was reasonable at all. So we said, well, we'll have a third-party life cycle analysis done on our process directly compared to a chemical process. Okay. So you go to a dye house, a real functioning dye house, and do the LCA. The really great thing about our process is most of it you can read straight off a meter. So, these are hard numbers. It's no longer handwave. Well, this is how much, carbon stuff you're saving by some algorithm that you have no idea how the data are inputted, right? So in this case, it's just reading it off the meter. It's like, how much electricity did you use? How much gas did you use? How much water did you use? So we looked at different fabrics. We thought, well, let's just test it against the one that's most environmentally damaging. So are you wearing polycotton, just out of curiosity? Probably. I'll bet your shirts.
You've mistaken me for someone who reads a label. No idea.
At least one of the two of your shirts is a blend of polyester and cotton. Okay. Do you have to iron your shirts, or maybe you don't bother ironing? I don't know. I don't.
I don't have to iron them.
I just put a jacket on.
You're talking to a man who washes his hair with soap, Jim. Who knows how he irons his shirts?
Anyway, so polycotton's good because you don't need to iron it, or at least not as much. Okay, but it has a really negative downside. You got to dye it once for the polyester and once for the cotton. So you're dyeing that fabric twice. Okay. Polyester dyeing requires a huge amount of heat to get conventional dyes into, and it requires a lot of water to wash it out afterwards. And the transition between dyeing the polyester and then dyeing the cotton, you have to treat in between, which again uses a whole lot more water. And the amount of energy and water that's used is really big. Okay, everything's a chemical, including amino acids in food, right? It's like protein. So our savings there is only about 65%. If you ask how many harmful toxic chemicals we save, it's 100%. But just on chemical savings, it depends how they're defined. In this case, it includes food, so it's not quite as big as you might imagine. But water, we save almost 80% in water, okay, for the reasons that I just explained. It's because the amount of water you need to run the process, then wash a lot of water, okay. And when you look at natural gas, the energy savings are massive because you don't have to heat up to really high temperatures. We only heat it up high very briefly just to inactivate the microorganism, okay. So it's like just sterilizing whatever you do. But that's the only reason. The dyeing process itself run at body temperature. It's not being run at 130 degrees.
You impressed the Earthshot judges. Anyway, you're a finalist in the end, a runner-up.
Yeah, well, I don't think we ever had a chance of actually winning in our category, but that's just me. So, but it was a good exercise.
And have you had any benefits from that?
Yeah, I mean, it's like everything else. You get introduced to lots of different people, lots of different investors. So it's been really great exposure from that standpoint. We've had pretty good brand interactions already, but even then you start, people that might have been not knowing very much about you, kind of hearing about you, and then they go, oh, really? Okay. And then they'll be much more engaged in trying to, partner with you to do a collection.
Yeah, great.
So yeah, it's been really useful because it is it's a global thing too. It's not just in Europe. And this year was in Singapore. And so were you there?
Our president and honorary secretary were.
Yeah, we don't get the good gigs. We get kept in this box to do these to the things.
Yeah. William, who normally co-hosts this with Lee, he was there.
Yeah.
So
Okay.
Orr was there, wasn't he?
Yeah, Orr was there, and our COO Chris was there because Orr had to. We do business whenever we can do it, and so he was there, and in Singapore, and go, oh, you got to go to India for a day for okay, well, so Orr went to India to do some business, and he came back. We needed somebody to cover while he wasn't there because there was lots of these other events going on, and one of them was kind of funny because Chris is really kind of a low-key guy for the most part, and so you don't really know a lot about his kind of background. He comes across as slightly posh, but you don't really know. Anyway, so they were going through the line, and Chris William was being introduced to all the people that were running the different companies. And he comes up to Chris and he looks at it and goes, "Chris, what are you doing here?" Because Chris was working for us and working the line, and so it was really odd to have this kind of thing where the person that's giving the prize knows the guys that are in the running for it, and
because they went to school together.
Yeah, they went to school together. So it's like you're always kids, right? I'm sure
it's a small world. It's always a small world.
Yeah, it's a very small world. So I think I don't think it influenced the outcome of the prize at all, but I think it has raised awareness, at least within the royal family we exist. So that's probably a good thing.
That's a good thing. It's a good thing. Jim, it's been lovely having you on the podcast. Thank you for kind of taking time out of your day to share your experiences and the story. Before we finish, though, we always try and have one sort of like tangential kind of question at the end.
Sure.
So bear with me on this one. It may or it may not work. I'll ask Neil first, then you, and then I'll give my answer. Okay. So I've been thinking, Neil, about fashion and colour, right?
Okay. I dread this bit of the podcast.
all the way through. No, it's a simple one.
Okay.
Without choosing a football shirt, okay, you're not allowed to choose a football shirt. What's your, in terms of fashion, what's your most favourite ever item of fashion you've had, and what colour was it?
See, I always get told off by my partner and my kids for my awful choice of
That was fair enough.
clothing.
Yeah, I go with that. Yeah.
I've got a very nice burgundy Fred Perry shirt that I'm very fond of. I've got a knitted one.
Is it very old? You had it for a long time?
I have, yeah. I've probably had it about 20 years, but I keep it as pristine as I possibly can.
Go on, Jim. Not that hat that you're wearing, that gorgeous red hat. But what's your favourite?
I think this is a good segue into what my favorite thing is. People should be careful about what they wear. They should think about what they're buying and not do fast fashion. Fast fashion to me is terrible. Of course, I'm old and grumpy. I'm older than you guys and probably grumpier than you guys. So I have a denim Levi jacket that I bought when I was 14 years old. So now it's over 50.
Oh, wow.
So it's over 50 years old. Thankfully, I can still wear it, which is pretty good. And I still wear it. It's faded and it's worn and everything else, but it's still serviceable. And sometimes I wear it to talk just to say, look, if you buy something that's decently made and you take care of it, it'll last you 20, 30, 40, in this case, 50 years, And so I guess that's also, it turns out it's a design icon too, apparently. I was talking to the former head designer for Levi's, and he was wearing one, okay, just same vintage as mine. I said, oh, I've got one too, only I bought mine when I was 14 years old and is in better shape than yours. But so it's one of those things. So yeah, I guess that would be my answer.
So I'm a bit of a shoeaholic, aren't I?
I knew it was going to be footwear related.
Yeah, I do like my shoes, and I'm quite known for my shoes. And I've always liked shoes. And this goes back to being 14, 15. I love winkle-pickers, Jim. I'm a bit of a kind of winkle-picker wearer.
You're serious?
Yeah. No, I love winkle-pickers. Okay. But I've never been able to get an exact copy of a pair I used to wear when I was like 15, 16 and in bands and stuff. They were very.
Do they have leopard print on
them? No. They were very winkle-pickery in that they were very long and very pointy. And they were bright yellow with a black lace thread running through them. And do you know what? I've been able to find almost the exact same style of shoe. And I've tried I keep buying white ones. I keep trying to dye them yellow, but I've never, Jim, managed to recreate that pair of bright yellow winkle-pickers.
You've come to the right man. Jim, what should he do?
You can't dye leather easily. because
I've discovered
it's made to dye before it gets dyed.
Yeah.
So if you try to dye over it, it doesn't work.
So that's it. I'm never going to recreate that great fashion statement. Then.
Well, you might if you can convince somebody to make a pair for you.
Yeah, that might be out of my reach.
Even
For me, that might be too expensive. Jim, it's been lovely having you on. Thank you so much.
Thanks a lot, guys.
Oh, Neil, you won't forgive me if I don't say to everyone that's listening, if you've enjoyed the podcast, give us a little review, because your reviews help other people find us. And, there will be people from the fashion world listening to this one, won't there? So
hopefully, I hope so. Yeah, if you're listening on Apple or Spotify, it does help if you leave a review, apparently. So we'd be very grateful.
Cheers, Jim.
Thank you.
Sure. Okay.
Great to see you.
Care, guys. Okay. Bye.